Hey, can you please switch on your cameras and can you hear me and see my screen? Okay. So, can someone please respond? Can you hear me? Okay, can hear and see. Okay, thank you. Right. Let's get started. And yeah, where are the others? Can you please switch on your cameras? So there are still people who don't want to show their faces, right? So you have promised someone that, okay, so throughout the lecture, I won't show my face to anyone, that kind of promise. So what? Hmm? So in fact, I'm not concerned about wanting to see your face, but I want to see some live students are behind the screen, so I can actually uh, do this discussion properly. Right. So just before sorry about yesterday, I had a power cut, and today exactly at 5:30 again, power went off. I think then today is also going to be a disaster. But it came back in 2 minutes time today. Right. Okay. So. Just before the end of the last lecture we had, I was introducing you the idea of super secondary structures. Right? Uh, we Now I think even though you are not coming from a proper background in biology, so now you have some idea about this protein structure, where we started saying that the proteins when they are made inside the body initially at the beginning it looks like a chain and it has different types of amino acid. These are the building blocks. There are 20 odd amino acids there and they're composed of only 20 different amino acids, but their sequence can be different from one protein to another. So that is why we can make virtually unlimited number of proteins with limited number of building blocks. So we keep changing the sequence. Then we said the tertiary structure. So what we call the after protein falls into a three-dimensional structure, we call it a tertiary structure, and that is the functional form of most of the proteins here. And this is called the tertiary structure. And in simple words, this is a three-dimensional functional form. So in between this primary structure or the sequence, primary structure and the sequence, and the three-dimensional tertiary structure, we have what we call secondary structures. Secondary structures. And there are two main types of secondary structures, alpha helices and beta sheets. I think we have heard these names enough times now to just to recall it, right? So the alpha helix is like a spiral and beta sheet is when the wire, the main peptide chain falls into sheets like this. Right? And here, um, the yellow one over here is an alpha helix. Now, these secondary structures can also get together to give you super secondary structures. Super secondary structures. That means it's an aggregation. You know the meaning of the word aggregation, right? They get together, right, to form super secondary structures. And then there are different super secondary structures, and these super secondary structures are given a special name. They are called motifs. Motif. for single, motifs for plural. And there are different types of motifs depending on what kind of secondary structures you have in them. So for example, now we have beta, alpha, beta motif. So this one, you can see. So initially there is a beta sheet, right? Then you have an alpha helix. Then you have a beta sheet again. So this is called beta, alpha, beta motif. And there's another one called Greek key motif. So in this Greek key motif, there are 4 beta sheets, but they are in different orientations, right? So you have one beta sheet going down this way, right? We can, from the starting and the ending point, this arrowhead is determined. So these two are anti-parallel. These two are anti-parallel, and these two are anti-parallel. So beta sheets, 4 beta sheets, oriented, organized in an anti-parallel way is called a Greek motif, right? And there are, if you have beta sheets like this, it's given a beta mean motif. So there are different types of names for them. Now, what is important about the supersecondary structures of proteins? They are very important features in the structure as well as the function of the protein. So in bioinformatics, what we often want to do is to identify these supersecondary structures in a given sequence. So we have to now come up with algorithms and methods to identify supersecondary structures of these protein sequences, and we call this activity specifically, we call it called protein motif identification. Protein motif identification. Right. So here's some definition on what I just said. So they are important in proteins to interact with other molecules, and then they are very specific, right? So specificity in protein interaction is a key. So you have to know that in biology, certain molecules only need to interact with some other molecules also, only with certain types of molecules. Not everyone is talking to everyone. Not everyone is interacting to everyone. So because there is a signal transduction pathway, right? What we call in the body. So for example, in the signal transduction pathway, proteins involved in one particular pathway are interacting only with specific types of proteins, right? So messenger, right? So there's a protein 1, protein 2, protein 3, likewise. So if this is a signal pathway inside your body, let's say as I explained last time, if this is the pain signaling pathway, so P1 will only interact with P2, but not with P3. So they are very specific. To maintain that specific interaction, these protein motifs are also important. Of course, they play a big role as a structural feature, right? Um, so usually these motifs often determine the biological function, right? Because, for example, now we have recognized motifs in certain classes of proteins. For example, there are a class of proteins, and these proteins bind to zinc metal, zinc, right? So then in these proteins, we have identified a special motif, a supersecondary structure called zinc binding motif, likewise. Okay. So it's always related to a function. So it is very important for us to be able to determine these supersecondary structures. Another particular, very specific motif is a helix, turn helix type of motifs. Now you can understand You can, if you look at carefully the picture on the right-hand side, the helix, you can consider this entire helix as a barrel, right? So it looks like a barrel, even though it looks like a spiral. Overall, it looks like a barrel. So here we have a barrel, right? So blue one over here. And then we have a turn, small piece like this. And then you have another barrel, right? So this is called helix, turn. This is the turn and helix motif. And these motifs are very important in binding to DNA, right? In binding to DNA. So we discussed that at the beginning. Our DNA is also like a double, this is called the double helix strand of the DNA. Now you imagine what is the structure of DNA look like? You take a ladder. Have you seen a ladder? How many of you have seen a ladder? No, you won't believe some students nowadays, they have not seen actual ladder because they are living in the virtual mm, environment. Have you seen the real ladder? How many of you have climbed on a real ladder? Right? So now if you have a ladder like this, good. If you have a ladder like that, and then you have the steps in between like this. So you take the ladder, and then what happens if you twist it, right? So if a ladder is flexible, if you take a ladder and then twist it, hold it from two corners and then twist it. And then you get the structure of DNA. Now you can imagine how it's going to look like. Then you have your ladder, the two sides like this. Then you have the steps in the middle. Right? This is how exactly the DNA double helix looks like. You have the connections in between to keep the two sides connected. Now, why it is important to have certain proteins to bind to DNA? Because proteins need to sometimes check the errors on replicated DNA. There is a huge mechanism happening inside the body, right? When one cell divides into two cells, you need to make a photocopy of your DNA material. Otherwise, what happens? Then you become something else, not a human, right? So your cells need to follow the instructions given in your genetic material. So genetic material needs to be duplicated, copied, intact, without a mistake. So in this, during this copying mechanism, for example, and after the copying mechanism, there are proteins come and read the entire sequence. These are happening at molecular levels. You can't imagine how complicated the nature is, right? You make a photocopy and then there is someone to proofread it. What is the someone? Someone is a protein, another molecule. Will identify if there are mismatches. Okay, so, and then these proteins, common structural motif in DNA, binding proteins, right? So, and then there are two helices. The first one is called the stabilizing motif. So that basically get hold of the DNA molecule. The second helix is called the recognition helix that interacts with the DNA and recognizes the DNA sequences, right? And crucial for recognizing and binding of specific DNA sequence. Sometimes it's looking for particular DNA sequences. Sometimes it reads the DNA sequence, right? And then for copying purposes and make sure that there are no errors. Now, okay, so I'm not going to go through into mechanism of DNA binding and all that, but we will get into now method. How do we look at these sequences and try to identify um, these motifs? And before that, we would like to, from the biology point of view, I would like to tell you two things about getting these um, motifs, right? Evolutionary, conservation, and functionality. Now, we say that motifs are highly conserved across different species. What does that mean? Some motifs, some of these specific structures like helix, turn helix are highly conserved. What does that mean? They don't change over the evolution. Highly conserved. Across different species, you will see the same type of secondary structures in different species, in maybe mammals, in other types of animals, right? So that means it indicates the fundamental roles of these motifs in the biological process. So that means you can't change these fundamental biological processes. So therefore, you must keep them. For example, now let's take a simple example. You want to modify your car or a bike, right? So you started with a scooter, small scooty, and then you want it to look like a Harley Davidson. The people do that, right? Okay. So you keep modifying things. But there are certain things that you can't modify, right? Because that is essential for the functionality of the bike, the scooty, right? So maybe your scooty looks like Harley Davidson at the end of the day, right? But for the scooty itself, there are certain fundamental, let's say engine parts, electronic components. These things are required. When you change them, it won't run. So it's same here with the genetic material, right? So the genetic material, when the animals evolve, they change from one species to another. They evolve in different directions, but there are certain fundamental units that you must keep. Why? Because you need to do certain important tasks as a biological creature, right? So that is what you mean by they are conserved. Right? So conserved things are not changing. Now, so there are two types of evolutions that you can think about. Right? So now I'm not going to go into too much of biology, but you need to understand because you need to get the idea of this so you can start thinking about algorithms and methods a little bit later, right? Um, the first one is the divergent evolution, right? Divergent evolution. So we start with the common ancestor, right? Common grandfather long time ago. And then what? Evolve into different species. For example, divergent evolution occurs when two or more proteins that share a common ancestor evolve and accumulate differences over time, leading to new functions or structures. Despite these changes, what does it say? Certain features such as specific motifs may remain conserved. Now, here you can see in this picture, I have put a common ancestor. It had a certain set of proteins. Then from the common ancestor, there were two species evolving into different plants, elephant and the mammoth. We have uh, we have seen the elephants, but we haven't seen the mammoths. I haven't seen, at least maybe you have. We have seen it in the Ice Age, right? Mammoths. But now these two animals got certain something in common. So their proteins are preserved because they need that function. And these small parts, these sequences in their protein are also preserved, right? And in the context of protein motifs, divergent evolution implies that the proteins in the sequence may have originated from the same ancestral protein. This ancestral protein contains a specific motif such as a structural or functional feature that remained unchanged despite the divergence of the proteins over the evolution of the time. Now, our one objective is to identify, bioinformatics is to identify these motifs and then to see where this motifs is related in terms of evolution. So in order to do that, of course, we need to use other tools such as phylogenetic trees and all that, right? And then we'll get into that. And then in this, if you think about now these motifs as patterns and then their divergence evolution, this is the part that is more related to you, the one on the right hand side, right? So you have a pattern, right? In your amino acid sequence, I would say that pattern is this, this, and that. You have a protein, but in that protein, you have 4 circles like this. Now that in that original common ancestor means grandfather, we had that structure. Then you started evolving into an elephant and a mammoth. But in the elephant, you still have this part. But it is a different protein now altogether, because if you think about the overall one, right, it is different. Why? You think carefully, look at it carefully. Here in the ancestor protein, original protein, this one. Original protein has a pattern of 4 purple dots. This one, now it has given you a different protein, this one, P1. Then that has 3 purple dots and a blue square. It's a different protein, it's a different sequence, right? On the other side, in the mammoth case, let's say this is the mammoth one, this is P2, the second protein, also coming from the original one. Now it has evolved, right? So it has got 2 different proteins from that. One is got 2 purple dots and a diamond and another purple. This has no relation to this one, no patterns here. But this another protein evolving from that in the same animal is got 4 purple dots, wherein once again you see this part is common. This is a pattern now preserved over these 2 species from the common ancestor. 3 dots is a pattern that is preserved over evolution. Right? Now our job is to, when you are given a series of dots and squares and diamonds, as from the software point of view, what are we supposed to do? We are supposed to identify where this 3 dot pattern is, right? So that is exactly what we are after. So we have the sequences. Now this is a particular motif. Now we want to identify this particular motif. So that is the objective in summary, right? Now in the divergent evolution, sorry, the convergent evolution, the other one. Divergent is when we start from a common ancestor and then branching out to different species. The convergent evolution is different species, animals started from different ancestors. They have no common ancestor in common. And then what? Right? Convergent evolution occurs when proteins that do not share a common ancestor independently evolve similar features or motifs. They have no common place to start off with. They started from different ancestors, but still over the time period, over the evolution, they have some common features or common motifs in their protein. This happens because these features provide a significant structural or functional advantage. So now you know why it happens, because for now, the example that I put here is for all 3 different types of flying creatures. You can see one is a hummingbird, another one is a bat, and the other one is a butterfly. They have no common ancestor. I mean, bird is a bird is a bird, which is the original grandfather, grandfather is a reptile, according to evolution theory. Right? So, bat is a what? Is bat a bird? Let's see how much of biology you know. Is bat a bird? How many of you think a bat is a bird? Hmm? How many of you think, it is not a bird? Bat is not a bird, right? So every flying thing is not a bird. That is the lesson that you learned today, right? So it's flying thing. It's a what kind of animal is that? It's a mammal, right? It sucks milk from the mother. It's a mammal, right? And then every swimming creature is not also a fish. You have mammals in the sea also. Who are they? Mainly dolphins and whales. And they are mammals. So here we have a butterfly, an insect, a mammal, and a reptile or a bird, actual bird. Now they started from different starting points. They evolved as individual creatures. But with the time, what happened to them? Look at their protein structures, right? So we have something in common. So we started with, let's say, this is a bat, and this is a butterfly, two of them. And then as they evolve, we will see now there is a common pattern. This is their starting point, and then both of them share now this common pattern. But this has nothing to do with the previous one, is it? But none of them had, right? None of them had this three purple dot pattern in their sequence at the beginning. Over the time, they developed this. So maybe it's a protein that is related to their flying function, right? So there are proteins that they have in flying creatures, but not on the animals that do walk on the ground, because flying is a different activity that needs different kind of energy supply, that needs to keep some strong muscles, but like keeping them light and all that. There are different requirements. So they need that particular protein, so they preserve it, right? It doesn't matter whether you are a bird or a butterfly or a bat. If you are flying, you better have this because it's a good thing to have when you're flying, right? So that's the basic idea. So this is the um, convergent evolution. All right. So these motifs can be mainly grouped into three categories: alpha, beta, and mixed. And there are different. This is just a picture to show you another set of different motifs, right? So a motif consists of only of um, alpha helices, um, beta sheets and alpha helices both, beta sheets only, and then here what we call a super role, right? Um, so where we have many sheets and barrels here into a specific shape, and then the barrel structures, and this is called the tin barrel structure. So there are different, these are the motifs that we come across usually. Right. Now let's get into computational techniques for identifying motifs. So this is the most interesting part. Now you know what a motif is, you know why they are important, and you know where they are coming from. And then you know why some animals want to preserve it. And then you know the significance of it. You know why we want to identify these motifs, because they play a structural and very important functional roles. So identifying the motifs can help us to identify the function of the proteins. Let's say you have isolated a new protein, maybe from humans or maybe from some other animals. It's a brand new protein, right? Now you do the sequencing. You isolate it. You do the sequencing. Now you have the sequence in front of you, but you really have no clue about what this protein is doing inside the body of a human or some other animal. Then you can do this exercise and then try to identify the common known motifs in this new protein and then kind of try to predict its functionality, right? So then predicting the functionality leads you to uh, identifying its role very well. So you think it is doing, helping in flying, then you go in that direction and do more investigations on that, right? So that is how people get to know the structure and function of these new proteins day by day, right? It's very important. Now, computational algorithm. So, methods. Sorry, right. So right. Thanks. Um, Right. The first computational technique of identifying the motifs is the sequence alignment, right? Sequence-based techniques, sequence alignment-based techniques. The first one is the MSA. We call it the multiple sequence alignment. So what is the purpose? Align several sequences together to identify regions of similarity, which may represent conserved mode. So in this case, we are not looking at any three-dimensional structures, even though these motifs specifically refers to the secondary structure, right? Secondary means they are already 3D structures, right? Because now, don't say that they are not 3D, right? Because this is the chain. When I make a alpha helix like this, a coil spring type of thing, it's an alpha helix. Is this a three-dimensional structure or not? Sorry, it's alpha helix. Is it a three-dimensional structure or not? Even though I call it a secondary structure because this is the secondary structure. This is also a three-dimensional structure, right? Yes. So, but in the sequence alignment methods, we are not looking at any three-dimensional structures, not taking the spatial arrangement of the sequences, just sequence-based. There are tools to do that. The first one is the Pastel Omega algorithm. So widely used uh, tool in multiple sequence alignment. I think this will bring you there. Yeah, this is the website. Now, what I would like you to do is to develop websites and standalone programs and apps like this. This is your ultimate objective. This is at least the ultimate objective of this lecture series in bioinformatics, which is taught to the software engineers, right? To be able to design tools like this. After understanding the bioinformatics, how can you contribute? How can you solve to contribute? How can you contribute to solve the problems in bioinformatics? Now, this is one such tool, Cluster Omega. So here you can paste your protein, DNA, RNA sequence, right? Paste your sequence here or use the example sequence. Now, what I'm going to do is I'm going to choose the example. Now, how does my example look like? So it's a protein sequence. Now I want to compare this one with three other sequences at the same time. This is why it's called multiple sequence alignment. So I want to compare human hemoglobin subunit alpha, whatever protein subunit from hemoglobin. It's a protein. We all know what hemoglobin is, right? It's a protein that helps us to take oxygen from one place to another in the blood. It's hemoglobin, right? So that's you also take the in your blood tests. You also take the hemoglobin count to see if you have enough hemoglobin. Right? And then I want to now compare um, this particular subunit of human hemoglobin with mouse hemoglobin subunit to see if what is the purpose then. I want to see if mouse and human in terms of hemoglobin protein structure, do we have any common features, common motifs, right? And there's this guy. Hemoglobin subunit of, don't know the biology name, let's search for that quickly. Because it must be a different species. Ah, Capra herpes means goat. Capra herpes means goat. Now you can put a protein isolated from a Sri Lankan politician and then compare it with donkeys, buffaloes, goats, foxes, and all kind of notorious animals in the animal kingdom and see if they share any common features. Only the Sri Lankan politicians, not us. Okay, so now to understand if there's any similarity. Now let's see, okay, all sorted. So we are comparing human hemoglobin with mouse hemoglobin and goat hemoglobin. Now these are different algorithms, right? So these are different algorithms and formats, right? Output format for in which way you want, sorry, these are output formats. They want the output in a specific way. In the master W, there are specific ways. So we keep it in the standard. Call it just, and then just submit it. You see how efficient these algorithms are. That's it. It didn't even take, but 20 seconds, I don't think that. So results. What are the results? Alignments. So here are the alignments. The three sequences, human, mouse, and goat. So there are different ways of, there are phylogenetic trees. Yeah, you can download the results files, right? So you know exactly. So there are certain matching areas you can see. Right. Now then, what is the next job? So the biologist is to take this matching uh, regions and then interpret them, right? So that's up to the biologists. You are not going to do that. We are not going to do that, right? So what we are worried about is how to get this job done. What is the job? You've given one sequence and then comparing it with two more or multiple sequences and identifying the regions. And then give all the details, how many as a percentage and all that we have spoken about this. Then they will try to interpret. So you get the blood results, you go to the laboratory. Their job is to do the sampling correctly, prick your finger correctly, and then get the count on a piece of paper. Then you take this report to the doctor. It's up to the doctor to interpret the results and then come to a conclusion, do a diagnosis and conclusion. So that part is someone else's job. So what is our part? Our part is to come up with the algorithms, the computational techniques, right? So this is the multiple sequence based methods. Now, let me go back to my original slide. That one over there. And there's another server called multiple sequence comparison by log expectations with different algorithms, right? And how these things work? By aligning sequences from different proteins, researchers can identify regions where the sequences are conserved across different species or within protein families. These are conserved regions often correspond to functional motifs. They are conserved, they often. Now, if you want to actually see, now since we have done certain uh, activities also, if you want to see if they are really um, corresponding to secondary structures or not, what do you do? What should you do? You can take these three sequences. First of all, see if you have there are three-dimensional structures available in the databases. Now, what are we trying to do? Now, after doing this multiple sequence alignment, the result showed us there are certain regions, they are what? Conserved and they are identical. Now we want to see if they correspond to actual secondary structures. One thing you can do is if they have the 3D structure determined by experimental technique. So you have the actual file in the PDB. So you can download all three structures and then look at these matching regions and then see, okay, here you have the ABCD sequence and that belongs to an alpha helix, alpha, beta, alpha motif in human. So how about that in the mouse? Is that also in a alpha, beta, alpha motif? Then we look at the protein from goat in the 3D structure. Is it also there in the alpha, beta, alpha motif? So if you have the structures, 3D structures, you can load it up onto a visualization program and then do a visual inspection. One thing you can do. Otherwise, you take the three sequences and then you can model them, model the entire protein using a server like AlphaFold, and then do the same exercise, right? So that way you can try to identify the motifs. Right. Yeah. So in multiple sequence alignment, one other tool that you have to be aware of is the position-specific scoring matrices, right? So this matrices, it's a kind of scoring method, quantify the conservation of amino acid at specific position in a motif, right? So, for example, um, in the alpha helices, in the known alpha helices, and in the known beta sheets, uh, let's say alpha helix, beta sheet, and an alpha helix. This is a particular motif. Then you have about Let's say you have 20 amino acids, right? So, for example, sequence, let's say ABEF sequence. ABEF sequences, this sequence is found in almost all alpha, beta, alpha motifs. Now, if you scan the entire database of structures you have, then you identify all the main motifs. You can also generate a database of motifs, common motifs, right? Let's say you have 500,000 three-dimensional structures in the protein data bank. What do you need to do? Right? You need to go through them and then identify these types of motifs and then class them, group them. Beta only, mixed, alpha, beta, and anyway you can, in either way you can class it like this, classify it like that, or classify it like this, right? Any classification way you can classify it. Then what do you do? When you are here, at this particular point, we can see that the sequence ABEF is commonly found in alpha, beta, alpha type of motif. Now then, for this reason, you can use a scoring matrix, position-specific scoring matrix. So if this sequence is found, we want to quantify. So if it is found here, we want to quantify the presence of ABEF here. We want to give it marks, right? So in the alpha helix, in the alpha, beta, alpha motif in this particular unit, if this sequence is found on the alpha helix, so that is good because that is the normal trend. So we can give them marks, score them, right? So you can use this kind of scoring method to verify whether you are seeing this kind of structure or not. Now, because what you are looking at is here in the sequence, we are not looking at any secondary structure, but you are looking at the sequence. So somewhere in the sequence, you have ABEF, ABEF. Now, ABEF, from our past experience, we know that this is most found in alpha, beta, alpha, the motifs. So we give them scores. That's it. So each of them is given a, let's say, 1 mark, right? And then next, this another sequence, GHFA, is found on the beta sheet. And we also see that in our sequence. Right? So this is from the database. This is in our sequence. Okay, so if this is also seen, it's good, right? So it looks like, so for this, now this segment so far looks like it gives you the prediction ability by scoring them to say that it's a alpha, beta, alpha type of secondary structure or the motif. So scoring matrices are important. Position-specific scoring matrices are important. Based on the position where they are, we try to give them a score, a mark, right? So thereafter, at the end of the day, you can calculate the total mark that you get and then come to a conclusion what kind of secondary structures you have in the sequence. So these are the sequence alignment based methods. Right. Now we come to a more interesting algorithm called hidden Markov model. These are called profile-based models. Have you looked at hidden Markov models in your database in your algorithm class? These are not used in bioinformatics only. Hidden Markov models are used in various types of problems. Have you used the hidden Markov models before? Yes, no, don't know, can't remember. Huh? What is happening? Just looking at my time. Okay. So then let's look at the hidden Markov models. So hidden Markov models, after the name Markov, so think of Russian statistician, develop these models and algorithms. So hidden Markov models, so HMM algorithms are almost everywhere, right? So you can use them to come up with different solutions to different types of problems. The models are statistical properties of conserved moieties within protein and families, right? So, so what is the basic of the hidden Markov model? Let me see if I have, yes. Now, here's a kind of a simple example. In the hidden Markov model, there are states, right? Um, States are there and they are hidden, right? You can go from one state to another. And then going from one state to another is probabilistic, all based on probability. Now, let's look at this entire scenario is whether a person going to go on a walk, on shopping, or to do some cleaning, remaining inside the house, depending on the weather condition. You have two states. The weather can either be rainy or sunny. You start with a certain condition, right? Now, the possibilities, I must tell you that the probabilities, the numbers that you see on this diagram, zero point 0.6 and zero point 0.4. When you start, you're going to go into rainy state is a 0.6 and you're going to go into sunny state is 0.4. That's how you read this diagram, right? You have the starting state, you have the inner hidden state, and then you have the uh, observations or activities, right? These are the three layers of hidden Markov models, and then also other components, the states, the observations we said. The initial state probability is also another component in a Markov model, and then transition probabilities, right? Transition probabilities from one state to another. Now, the next question come to your mind is how do we get the probabilities? These probabilities are based on what we call the previous training calculations. We use a training set data, what we call the training set, to look at this probability, right? So this is how you train your model. This is exactly what we sort of do with the artificial intelligence, right? You come up with an algorithm, you need to train it. So to train, you need to have some data. So the data that you use to train your model is called the training set, right? So larger the training set, better the training is. Okay. Then better the training, better the prediction power of your model is, right? So this is how this entire thing works. Now let's look at, first I take this example, then I'm going to uh, give an activity for you to do, right? Um, So we'll come to the motif recognition first. Now component states. This is the actual application. Okay, so let's look at this now. What are the states we have? The rainy, one possible states of the system. Okay, sunny, the other possibility. The model transitions between these states over time. So then you can go to this part here and then look at the transition probabilities. So going from rainy to rainy is 0.3. Is 0.3. That's what this arrow means. Going from rainy to rainy. The next one is going to be also rainy day. Is going to be a 0.3. Going from rainy to sunny is 0.3. Going from rainy to sunny to rainy is 0.4. Going from sunny to sunny is 0.6. And then if it is on a rainy day, going on a walk is 0.1, the probability. All the probability should add up to 1, right? In probability theory, there's nothing bigger than 1, right? Probability is equal to 1 means it's 100%. There's no other option, right? So based on this 0.1, for example, on the training day, so we have looked at enough people on a rainy day. If you look at 100 of them, how many of them will go on training, on a walk? To get the probability of 0.1, how many? On a rainy day, if you look at 100 people, only 10 will go on a, what? 10 will go on a walk. So 10 out of 100 is 0.1. That is the probability. So this probability is calculated based on your training set. So not only you can't just look at 100 people. When you do this training, you have to look at, let's say, about 100,000 people to get a reasonable probability. You have to have a very larger training set, right? So that's must in the algorithms where you have training set, you have to have a very large data set to train. Otherwise, your outcome is not good. That is what ChatGPT is basically learning from user response also, continuously learning, growing, right? So its data set its training set also becomes what your user response become part of its training set. So it has a ever growing training set, right? So that is how these machine learning models work. So this is something similar to that. So then go on a shopping on a rainy day is 0.4. Go on do cleaning, staying inside the house is 0.5 on a rainy day. So 0.5, 0.4, 1 adds up to 0.1 adds up to 1 on a rainy day, right? This is 0.9, 0.5 plus 0.4 is 0.9, 0.9 plus 0.1 is 1, right? So likewise, on a sunny day, 6% of them go on, sorry, 0.6 is the probability to go on a walk. So that's what the red dash line means. On a sunny day, going in a walk, probability is 0.6. On a sunny day, going on shopping is probability is 0.3. On a sunny day, doing cleaning is 0.1. Now, how does this thing work? Right? So initial state, the person is more likely to start in a rainy condition with a probability of 0.6 compared to sunny condition with a probability of 0.4. In this particular setup, when you start, there's a higher probability to fall into a rainy day because this is 0.6, this is 0.4, right? If today is rainy, there is a 70% chance that tomorrow will also be rainy and 30% chance that will be sunny. While this is 7.7. Rainy is 0.7. If today is rainy, next step going to be rainy, next day is going to be rainy is also 0.7. So going from rainy to sunny is 0.3, only 30%, right? If today is sunny, there's a 60% chance that tomorrow will also be sunny. So if you roll into here, 40% or 60% chance tomorrow is going to be sunny, and 40% chance tomorrow is going to be rain. If the person is most likely to stay inside, okay, if it is rainy, the person is most likely to stay inside and clean, the 50% chance is there, right? So now, how do you use this to do a prediction? How do you use this to do a prediction, this model? Can someone explain it to me? Now you know how a hidden Markov model works. How does it work? Now I want to see tomorrow. Today is rainy. And I want to predict what I will, instead of knowing the inner states, I have no idea about the inner states, right? What I know is today is rainy, and then I will have three outcomes to choose from. Then the prediction is done by the Markov model, right? So it will lead me to the rainy. Then what will happen? If today is rainy, tomorrow is going to be rainy is 0.7. If today is going to be, tomorrow is going to be rainy, then you will be doing one of the rainy activities. Then the rainy activity, which has the highest probability of doing is cleaning, right? So then the model will tell you, okay, if today is rainy, most likely you are going to do some cleaning, staying inside the house, right? The user has no access to the middle layer, how it works. It's hidden, right? So you have to develop that model, okay? So looking at the training set, looking at the data that you get, look at the probabilities that you calculate, you have to develop the hidden Markov model. Now, okay, so how could you use this to identify the protein secondary structures? Secondary structure elements, such as what are the elements we have? Alpha helices, beta sheets, and random coils. Right? Mainly in super secondary structures, you have alpha helices, beta sheets, and then random coil like this. So these are the main three features that we have. And these are not directly observable. You can't observe these things. What you observe is a sequence of amino acids, right? So the observed sequence is the sequence of amino acids. For example, you have the ports A, B, C, D like that. Each amino acid has a tendency to appear in particular secondary structure. But this is not deterministic. This is probabilistic. This entire model works on the probability. Let's say you have an amino acid, let's say B. So B can, so then when I say it, it's not nice. Let's say amino acid P. So you have A, B, C, D, like that 20 of them. P. P can be here also. P can be found in random coils. P can be found in alpha helices. P can be found in beta sheets. But from your analysis, before you model this, build this model, from your training part, you know that P has a higher probability finding in the alpha helices than in beta sheets. That, of course, a known factor to you. From where? From your training part, right? So P has a probability of, let's say, 0.6 of finding in an alpha helix, and it has a zero point 0.3 of finding in a beta sheet, and it has a zero point 0.1 probability of finding in a random coil. Now, what are the hidden states? Three states in this model: alpha helix, beta sheet, and coil. We can label them H, E, and C. The probability, there is a probability for each amino acid finding in each type of thing from your trainings, and then transitions, probabilities. This defines the likelihood of transitioning from one secondary structure, a hidden state, to another. For instance, there is a probability that alpha helix might continue or transition into beta helix. That can happen during the dynamics because it's a structure like this now, right? So it can, during the normal dynamics, you have an alpha helix, now it unwinds and then gives you a small beta sheet. There's a probability, but that probability could be very small, I don't know, right? So there's also a probability that a beta sheet can go into an alpha helix. Of course, there's a probability, high probability, for example, that we have seen in our simulations. These alpha helices, the small alpha helices like this, right, can go into loops, coils, uncoil, and then come back as an alpha helix. Uncoil, alpha helix. So these we have seen quite often, right? So there's a high probability. There are three states, and then you have different probabilities. So the only thing that you have to understand is that these transition probabilities and finding these amino acid residues inside each type of secondary structure, these probabilities are kind of pre-found, right? It's not found during the execution of the algorithm. You have these probabilities found beforehand. Now what is happening here? So the transition probabilities defines the likelihood of transitioning from one secondary structure to another. For instance, there is a probability that an alpha helix might continue into beta sheet. Emission probabilities. This defines the probability of an amino acid being generated or emitted from a specific secondary structure. For example, certain amino acids are likely to appear in alpha helix than a beta sheet. Right? So then all depends on the probabilities, observations, then transition probabilities, and then finally you say that your particular amino acid P is more likely to be in an alpha helix, right? So it's next. So what would be now you keep doing it for the next amino acid. Now you have a sequence that you keep reading. Let's say you have this part of the sequence. Now we are reading this part of the sequence, P, F, A, B, for example. Now, when you use this algorithm, your algorithm tells you amino acid P has higher probability of being in an alpha helix. Then you, okay, fine, higher probability. You read the next one. The model predicts that F also has a higher probability of being in an alpha helix. A also has the highest probability of being in an alpha helix. This also has a higher probability of being in an alpha helix. So what is your conclusion? So the sequence PFAB belongs to an alpha helix. So the secondary structure that is coded by this sequence is an alpha helix, right? So that is how these are probabilities, right? So then sometimes then there are these things, it's complicated than we think, but this is the simpler idea, right? Because then sometimes F is replaced in C with C, and F and C are sort of very similar in terms of amines. Then you have to use a replacement matrix to see if you're going to really penalize this or whether you're going to give full marks for having C instead of a F in an alpha helix, right? So likewise, then you can predict what's your entire sequence is responsible for, right? There's an same thing, amino acid sequence and okay. So this is the basic idea about the hidden Markov model, right? So in a hidden Markov model, what are the main components? The observations, states, and emissions, right? The outputs and the probabilities. Probabilities of what? There are two types of probabilities that you need to worry about. The probability of switching from one state to another and the probability of giving out one result from one state. These are the probabilities. So these probabilities, where do you get the probabilities from? We get the probabilities from our training data, right? So that's the hidden Markov model, all based on probabilistic. Right. So the next computational or the bioinformatics method that we're going to talk about is called the structural alignment and comparison. They are kind of um, different to sequence alignment, just pure sequence alignment, right? One specific thing that we can look at is called the distance matrix alignment, tool for comparing protein structures and identifying structural motifs. So before we talk about how it works, so here's a small video that we're going to watch on how this distance matrix method works. Okay, so then I'm going to share the video. Why don't you see the first order? I can't see the option to share the sounds. participants. Not complete today. Okay, anyways, let's start and see. If you can't hear the sound on that video, let me know. Okay, just put a message on the chat box. Okay, can you see the screen? See?
The next method for proving
Don't know. For this option, usually okay. Can someone from the institute, anyone here help me sharing the voice of this video, please? Usually on the Zoom panel, I have an option to share the sound. Today it is not visible. Um, Okay, I think I got it correct. Let me try it again. If you can hear the sound on this video, please let me know.
The next method for building phylogenetic trees that we will talk about is the group of methods called distance matrix methods. There's a range of different methods that are all based on what we call distance matrices. And I will explain the main ideas in that, and we will then look at some particular examples with some of these methods. So, what is a distance matrix? Well, the idea in distance-based methods is, as always in phylogeny reconstruction, in molecular evolution at least, that you start out with a multiple alignment of sequences. I've shown a small example here with just 3. From the multiple alignment of sequences, the idea is now that you should count what we call the genetic distances between each pair of sequences. The genetic distance between a pair of sequences is simply the number of mutations separating them. So in this particular case, if you compare, for instance, gorilla and human, you can see that there are 4 mutations separating them. There are 4 positions in which the gorilla sequence differs from the human sequence. So we now build a small distance matrix. A distance matrix is just a table showing all the pairwise genetic distances. And in the entry for gorilla versus human, we now put the number 4, indicating there are 4 mutations. If we compare human and chimp, you can see that there are 2 positions that differ. So human and chimp, we put the number 2. Finally, chimp to gorilla. If you compare it again, you will see that there are 4 locations, 4 positions, the last 4 in the sequence, that differ. So gorilla to chimp also has a genetic distance of 4. The other half of the distance matrix is, of course, going to be the same as the first. It's a symmetric thing, so you only have to fill out one half of this matrix. Now, the idea in distance-based methods is that at this point we forget entirely about the original set of sequences. We focus just on the distance matrix, and the goal is now to build a tree with branch lengths such that the distances measured along the tree correspond to the ones that we actually observed from the alignment, to the distances we have in the distance matrix. And what is a branch length, incidentally? Well, a branch length in a tree is a number indicating the number of mutations that have occurred on that branch. So in this particular example, that's a really simple problem to solve. We see that human and chimp have a genetic distance of 2. In this particular tree, I've put human and chimp closely together. There's a distance of 1 on this branch, a distance of 1 on that branch. So the total distance measured along the tree between chimp and human is 1 plus 1, or 2, which is what we also saw in the alignment. Distances measured along trees are called patristic distances. That's a term we'll use now and then. Let's just check the other distances. Chimp to gorilla.
Just hold on a second. The way that it plays is not so good. Let me see why it is stuck in a
Distance measured along the tree will If we compare human and chimp, you can see that there are two there are 4 locations, 4 positions, the last 4 in the sequence that differ, so gorilla to chimp also has a genetic distance of 4. The other half of the distance matrix is of course going to be the same as the first. It's a symmetric thing, so you only have to fill out one half of this matrix. Now, the idea in distance-based methods is that at this point we forget entirely about the original set of sequences. We focus just on the distance matrix, and the goal is now to build a tree with branch lengths such that the distances measured along the tree correspond to the ones that we actually observed from the alignment, to the distances we have in the distance matrix. And what is a branch length, incidentally? Well, a branch length in a tree is a number indicating the number of mutations that have occurred on that branch. So in this particular example, that's a really simple problem to solve. We see that human and chimp have a genetic distance of 2. In this particular tree, I've put human and chimp closely together. There's a distance of 1 on this branch, a distance of 1 on that branch. So the total distance measured along the tree between chimp and human is 1 plus 1, or 2, which is what we also saw in the alignment. Distances measured along trees are called patristic distances. That's a term we'll use now and then. Let's just check the other distances. Chimp to gorilla, the distance measured along the tree will be 1 plus 1 plus 2, which is 4, of course, and which is exactly what we have in the distance matrix. The distance from human to gorilla will also be 1 plus 1 plus 2, which is 4, and which is what we have in the distance matrix, which is the number that we observed simply by comparing the sequences. So this is the essence of all distance-based methods. Take a multiple alignment, count the pairwise distances, build a tree such that distances measured along the tree correspond to the distances you saw in the alignment. To put that a bit more formally perhaps, you have a set of sequences, you count all the pairwise distances. The goal is to build a tree where you place the sequences on various leaves. There are different possible ways in which these sequences can be placed on the tree. In this particular case, I put sequence 1 and 3 closely together. The goal is now to find the branch lengths. I've labeled them A, B, C, D, and E on this tree. To find the branch lengths such that the distances measured along the tree, the patristic distance, is as close as possible to the observed distance. The observed distance, we refer to that with uppercase D's in these slides, are the numbers we got just from looking at the alignment. The patristic distances, the tree distances, which we indicate with lowercase d's, are the distances that we find by adding the branches, the lengths of the branches separating those two sequences. So for instance, sequence 1 and 2, the distance between them measured on this tree is A plus B plus C. Sequence 1 to 3 measured on this tree is the same as A plus D. So every patristic distance corresponds to some sum of branch lengths. And the goal in distance-based methods is, as I say, to find these branch lengths such that the patristic distances are as close as possible to the observed distances. I'll return later on to why it's not always possible to make them be exactly like the observed distances. Sometimes it is, sometimes it isn't. At this point, you should take the next weekly quiz for this week. Try to solve it. You will, in this one, get the opportunity to try to manually do a distance-based analysis. I've given you an alignment. You're supposed to, from that alignment, first construct a distance matrix, simply counting the pairwise distances, the number of mutations separating each pair of sequences. Then, from that distance matrix, you should then construct a phylogenetic tree such that the method for building phylogenetic
All right. So that was a very brief introduction to distance metric methods. So what we want to do is we have two or more sequences. We want to see how close or how far they are from one another. How far in the sense with the distance means the actual evolution rate, the sequence similarity. Right? That's what you mean by distance. So I'm far away from you means we have less similarity in this sense. So that's what the term distance operates here in this sort of algorithm. Now, before we start going into the steps of this algorithm, I want to ask you, did you see that what that professor asked the student to do at the end of the lecture? Right? He wants his students to write the algorithm, to do this manually first, see if they can do it. Right? So build up a distance matrix and then do these distance calculations by hand before you automate, right? So this is the approach that I also take, right, in teaching this course to you. Why? Because at the end of the day, the most important thing is you understand the concept, right? You understand the concept, and then you should be able to come up with the algorithm first to solve the problem. Do it by hand first. If you want to take a small problem and do it by hand, then you can automate it, then you can scale it up, right? So that is always a good approach in designing these bioinformatics tools, because if you come up with a very large data set to start off with, and then you are trying to still develop the algorithm, it will be difficult, right? So take a small data set. If you are doing a sequence comparison, if you are doing a distance analysis method, right? Take a small data set and then see if you can do it by hand first. Then you know how to solve the problem. Then you automate it. Then you scale it so it can work on a large data set, right? So this would be a good approach. So the distance matrix method is basically what we call here is the distance metric alignment, or DALI. Is a widely used tool for comparing protein structures to identify similar 3D structural motifs. It compares the distance matrices of two protein structures to determine their structural similarity. So looking at the individual matching, finally, you should be able to see how these two big proteins are close or far away from one another. So that's what he tried to do in that example, right? So look at the genetic code, the, the sequences first, and then see how far a human is from chimp, how far a chimp is from gorilla, right? So that is the activity, that is the objective we want to do. We have proteins, three proteins, and then we want to see which two are the closest, and then what's the next two, likewise, right? So how it works? Okay, here distance matrix calculation for a given protein structure. DALI calculations. Okay, DALI calculates the distance matrix, where each element of the matrix represents the distance between pair of residues in the protein. Then the next step is the alignment. Then aligns the distance matrix of two proteins by finding an optimal superposition that minimizes the difference between their distance matrices. Then you have a Z-score calculation. The similarity between the two structures is quantified by using a Z-score. Like the Z-score is same as the Z-score that we use to select the students from the advanced level class. So what is the output that we expect? Outputs an alignment of two protein structures highlighting regions with similar 3D folds. So we take maybe two sequences to start off with, and then input is the sequence, and then how we do it is by calculating the distance matrix and then score them, and then final output is the matching regions, right? So let's take an example. So we have protein P and Q. Yeah, right. So we calculate now what we want to see how far uh, protein P is from protein Q, or in other words, how close protein P and Q. Before we do that, we have to generate individual distance metrics for protein P and protein Q, right, for each residue. So distance metrics here, the distances are on the branch like here. This is the circles here are called nodes, right? So here we have amino acid sequence in the protein P, 1, 2, 3, 4, 5. So here's the 1, 2, 3, 4, 5. Let me 1, 2, 3, 4, 5 here. These are the amino acids. And then we have a square matrix. It's like a, if you have 5 um, residues, it's a 5 by 5 table. That's what we call it, a matrix, right? 5 rows and 5 columns. It's 5 by 5 matrix. It is square, so that's the same number of rows and same number of columns. It's a 5 by 5 matrix. In this case, this is 4 by 4 matrix, right? And this is why I call distance matrix, because this table, the matrix, is used to calculate the distance, right? And the distances for the protein P. Amino acid 1 and 1. It's the same thing. There's no distance, so we call it 0. Right. So now I want you to understand how this distance matrix is generated based on this information in the graph. So amino acid 1 and 2. What is the distance? 2. 1 and 2. The distance is, distance is A. Right. So 2 and 1. Therefore, 2 and 1, therefore, is also A. Then 1 and 3. 1 and 3. It is B on this dash line. It's B. So 3 and 1 is also B. So likewise, we keep doing this. So you get the distance matrix for the first protein. Then you calculate the distance matrix for the second protein, right? Then we do what we call row matching scores, row by row matching scores. Now, what's happening here? Can someone try to explain what has happened here by looking at the numbers there? Yes. So this matrix compares each row from distance matrix of P with each row from distance matrix of Q. First row of P has 1, 2, 3, 4, 5, and then distances 0, A, B, C, D. Right? So when you compare this set with that distance, how many matching do you have? How many matching do you have? How many matching elements? 4. 0, A, C, and D are both in here, this row and that row. 0, A, C, and D. There's no B, so it's 4. So it's 4. Right? Second one, second row, and row A, the first row of the other one. How many matching? How many methods? Hmm? All right, did you figure it out? What's happening? Right? So I've done the first one for you, the first row with the first one here. For example, you take this row O, A, B, C, D, and then compare it with the first one over there, right? O, A, C, D. How many elements matching? 4. Now you take the first row and compare it with the row B. O, A, B, C, D, right? What is here? B, A, O, F, G, right? So then you put the matching scores into this matrix, the second. So you can do this as a homework. So you can write, what I want you to do is write a small code to compare these two matrices and then give them a mark. If you have 4 elements matching, make it 4. If you have 1 element matching, make it 1. Then what you can do in this algorithm, what's happened to next is to align them. So align P and Q. So we can say the first one is matching with row A. The first row is matching with the row A, this one. Row A means starting with this one. Second one is matching with B. There's no matching for the third one, right? And the fourth one is matching with C, and fifth one is matching with D, right? So this is the outcome. So you can finally see which part of one protein is matching with the other part of the matching scheme. Of the, I mean the matching, what part of the one protein is matching with the other part of the other protein. Okay. So first one, the first thing that we need to do is to calculate the distance matrix, right? So we'll talk about this algorithm in more details. I think we have to press for time here. In more details in the next class, which is tomorrow. We'll start with this one, and then try to sort of do it again before we get into the other technique, which is the template modeling method, and then that also look at a score, right? Now, all these methods, whether they are probabilistic or whether they are, now there are, if you think about this whole thing, you have 2 sequences. What we are trying to do, we are trying to identify similar segments in the given 2 sequences, right? And then we have sequence-based methods. Then we can have distance-based methods. So we first look at the distance matrix and then see how similar or distant these 2 sequences are. Then we can also have probabilistic methods. These are the main algorithms, right? You should have a fairly a good idea about these types of algorithms, right? So when I want you to write down, let's say there's a question in the exam, right? So here the sequence is given to you, and there's another sequence given to you. Write 2 different types of algorithms to identify common secondary structures in these 2 sequences, right? The question can be like that. So you can select one from sequence alignment, the other one from probabilistic methods, hidden Markov method type of thing, right? So this is the main idea. So I think we should stop here and then take this example again in more detail, um, and then try to do it carefully, more carefully tomorrow, right? So we'll start with this example again. So I think it's better to do this. So you can, meanwhile, you can take a screenshot or whatever. Okay, share the screen. Again, you can take a screenshot of this slide. See if you can do the matching scores. So then we'll stop here and then we'll see you tomorrow. Right. So today we are doing yesterday's lecture. So tomorrow 6 o'clock we meet again. Okay. All right. Any questions? Or shall I stop here? All right. So then we'll stop here and then good night to you.